So, antimicrobial substances that help protect our skin and our mucous membranes. So our skin, particularly, is salty at times, especially when we perspire. And don't quote me on it, but what little I know about chemistry and stuff, it kind of makes sense. Like, anybody ever make homemade ice cream? We're actually going to do this on Thursday. You put rock salt and salt. You put rock salt and ice on the outside. What does the salt do to the ice, though? It actually kind of melts it a little bit. But what it does is it creates greater surface area, too. There's this unique phenomenon. I need to look up what it actually does. But so salt helps with this cooling process for us. That's why there's salt in our sweat. So just like how you add salt to the ice to have it cool better for making homemade ice cream, the same is true, which is why salt is in our sweat. I believe for that cooling convection action. Salt, as we know too, can be a deterrent for microbial growth. Like, we purposely in the past have cured salted meats, right, to actually preserve it from microbial growth. We didn't do it because we're crazy salt people, right? We found that it was antimicrobial in action. The problem is if you eat too much salt, right, it causes problems. Because it affects osmolarity, right? And that's what happens to the microbes too, right? As we talked about this before, right? When we talked about controlling microbial growth, salt is inhibitory because it will cause the water to be pulled out of those cells and cause them to shrink up like little raisins. And it's called plasmolysis, but they actually don't lyse, right? If you rehydrate things like cured meat, they could also rehydrate the microbes that you shrunk up when you salted it. Remember, there's not any one good, perfect way to preserve your food, right? This is why we typically use multiple methods. But, of course, the salt of our skin is antimicrobial. But there are microorganisms that do live on our skin, right? So what does that tell you about them? They're salt tolerant, right? Most staphylococci are salt tolerant, right? Because they grow on our skin, right? So they can grow on mannitol salt agar, right? Which is a higher than normal concentration of salt. Lysozyme is an enzyme that degrades peptidoglycan. Remember, peptidoglycan makes up what cellular component of what organisms? The cell wall of gram-positive and gram-negative bacteria in general. But of course, gram-positives are more sensitive to this, right? Because their outer covering is probably peptidoglycan, where the outer membrane of gram-negatives will somewhat protect them from lysozyme damage, but not completely. And then what this enzyme does is it literally goes in and breaks the sugars apart in the peptidoglycan. Peroxidase enzymes actually produce oxygen radicals, right? These are highly damaging forms of oxygen. Lactoferrin, this is a protein that we produce that binds to iron. Iron isn't very soluble in solution, right? So the proteins help solubilize it. But also, if our proteins bind to the iron, it stops the microbes from being able to acquire the iron. And microorganisms, remember, need iron for their metabolism too, so much so that some of them have developed special hemolysins, right, that will actually lyse red blood cells to get at the iron in the hemoglobin. So remember the blood agar plates, that's why we look at those, right? They have intact red blood cells, and some of them produce hemolysins that will completely annihilate the red blood cells. What type of hemolysis is that one? Beta or alpha? Beta. Complete destruction is beta, right? You can see right through the plate. And then alpha is where it pokes holes in the red blood cells. And that one turns kind of greenish in the plate because the oxygen goes in and binds with the iron and discolors it. Defensins, these are molecules, small protein segments that can insert into the membranes and disrupt them. So it's like throwing little daggers, right, at the membrane and disrupting the membranes of foreign cells. It's not specific as to what organisms it's attacking, right? So what body fluids contain lysozyme? What you think? Our tears, our saliva, our mucus, our tissue fluid, and our blood. All of them. All of them. Right? Because they would all benefit from being able to digest peptidoglycan. And remember, that's unique to bacteria. So we don't have to worry about this enzyme digesting us, right? We're not made of peptidoglycan. Bacteria are. So as I mentioned earlier, right, our stomach is really good at destroying stuff. Its main function is protein digestion, and how it does that is the pepsinogen, which is an enzyme that gets activated by the hydrochloric acid that our stomach actually produces. Right? This is why stomach acid can be so damaging, right? When it bubbles up into the esophagus or when you vomit extensively, it can cause damage because the other parts aren't equipped to deal with that. You have an extremely thick layer of mucus you make in your stomach to protect your stomach from eating itself, right? So, but that really strong acid, that hydrochloric acid, helps denature the proteins, helps to form them and make them more accessible to digestion by the pepsinogen. And then when you go into the first part of your small intestine, your pancreas is actually attached there, and it dumps a whole bunch of buffers, alkaline buffers, to bring the pH back up to more neutral pH in your small intestines. It basically neutralizes the acid. And then it dumps more enzymes in for digestion, right? So it wants to digest sugars, wants to digest protein even further, nucleic acids. Your bile that comes from your liver that's stored in your gallbladder also squirts in at this point, and this helps break up the fats so that they can be digested and absorbed in the small intestine. So for the most part, most of your food is digested and absorbed in your small intestine. That's why it's so, even though it's small in diameter, it's really long, right? Lots of surface area for digestion and absorption. When you get to your large intestine, there's really no more digestion happening by you. Your microorganisms, on the other hand, in your large intestine will digest what you did not digest, right? And you can get some stuff, mostly vitamins, from that, right? And the microbes that are digesting the food, they produce vitamins that are beneficial to you. You're sucking the last bit of water out, right, that you may need, because you dump a ton of water in for digestion. Right, so you don't want to lose that through defecation. Same thing, urination. If you don't produce enough, if you don't drink enough water, right, your body's going to put the least amount of water in your urine. And that's why it'll be what we call concentrated, very yellow, and very smelly, right, because the wastes are very concentrated in it. If you're in an environment where there's no water, should you drink your pee? Absolutely not. Absolutely not, because your body will conserve as much water as possible and put as little as possible in your urine to urinate. So again, why are you peeing? What are you peeing out? Acids, maybe even bases, waste products, right? Stuff that needs to be filtered out of your blood and leave your body. There's a reason why you're putting it out. Don't put it back in. Right? What little water is in there is not going to help the fact that you're going to make your body do it all over again, right? Not to mention, right, if you don't have a bladder infection, it should be sterile, so you don't have to worry about that, right? But unless you're in a situation where you could distill what little water is in that urine, leave it alone. Right? Leave it alone. There's a reason why you're peeing. to remove waste from your body. So same thing with defecation, right? Some animals will eat their own poop, right? Because you don't completely digest everything you ate, right? So there is potential of nutritional value in that. And my dad's dog actually had a pancreatic problem where she wasn't producing the enzymes necessary to digest her food. So she literally was pooping out food, right? And then she was still hungry because she didn't digest it or absorb enough of it, so then she would eat it. There are other reasons why cats and dogs will do stuff like that. Again, to them, it still smells like food sometimes, right? Depending how rich of a food you feed them. But they might have a medical condition too. That's the thing. So you should always look into it. So that my mom had to buy like really expensive, like ground up pig pancreases and sprinkle it on her food so that it would help her digest her food when she ate it. It's like $100 a jar. It's like ridiculous. People can get these types of problems too. Usually more noticeable in them. But the other reason why cows and horses and things like that, the babies will eat their mom's poo too, and why ranchers won't stop them from doing that, is to establish the microbial environment in their gut. So the same is true for us. Like you have a certain microbial environment in your gut, and these actually help protect you because the bad guys, when they come along, they don't have any food to eat because the good guys are there and they're eating it up, right? They're your normal microbiota. Same is true for your skin, right, and your vagina. If things are balanced, then when the bad guys come along, usually the good guys have taken up all the parking spots and they're eating all the food. And so they competitively exclude the bad guys, right? The bad guys have no place to take up residence. But there's a condition that can happen called antibiotic-associated colitis, where people who have been on long courses of antibiotics, right, they've destroyed most of the normal microbiota in their intestines. So then there's Clostridium difficile. It's a spore former bacteria that's found in the environment, and you can be exposed to it. Like you could ingest it. Most of the time it'll just pass right through the system. But in this case, because there's no competition in the large intestine, it can germinate and take up residence. The bad news is this is highly pathogenic. It will literally eat your intestines. So one of the ways they're going to cure you is actually put poop back in the system, right? So what they usually do is a poop enema from somebody in your own household because you should have the same or similar microbiota in your intestines. Sounds kind of gross, right? But yet very important. So this is the problem too, right? Everyone's taking probiotics nowadays. I don't know many people who aren't. But which end are you putting it in? Your mouth. So what happens to most of those microbes? They get digested as if they were food. If you truly want to change your intestinal microbiota, you got to come in the other way, right? Enema. I don't know who wants to sign up for that. I'm like, yeah, I'm good. I just try to eat right and keep everything balanced down there. Right? So, I mean, you have to take, you'd have to take huge amounts of microorganisms orally to actually change the population of microbes in your intestines. Truly.
Is that the same for yogurt?
Yeah, same is true for yogurt. You just digest them. You're not going to change your population. But, I mean, they're not bad for you. They're not bad for you. But they're not doing what people think that they're doing. Not going that route. But these guys are really important, right? Because when they're not there, you don't have that level of protection. You can end up, and I'll tell you, anyone who's worked as a nurse, thank God I've never experienced it, like, when this guy is growing, the flatulence from it is unmistakable. The smell when someone has Clostridium difficile, they call it just C. diff for short. You know before you even walk, yep, she smelt it. She's got that look on her face and the head is bobbing. Right? It's very distinct. So, same thing. Some people can smell Pseudomonas aeruginosa infections. Not to mention, too, they tend to have the greenish tinge to them because that organism produces pigments that will discolor the skin green. But it also has a very distinct odor. For me, I think it smells like, musty, moldy socks that have been left in the locker room that you forgot about. But to some people, it's like that musty dirt soil smell, which makes sense because Pseudomonas actually will grow in the soil, right? And that's pretty much where it comes from. And this is why, like, people who have serious burns and stuff like that, they won't allow potted plants into the room because the bacteria could be in the soil. They won't let plants in, period. And it's also why they make you wear the little footies, right? Because any dirt you're dragging in on your feet could contaminate the wounds of the patient. So, yeah, it's all about protection, right? But obviously stuff gets past this, right? These layers of protection. So, as I said a moment ago, right, we have these competitive exclusion that happens that helps us by the normal flora being there, right? The E. coli are going to compete. Canidia albicans, even though it's in the vagina, it usually doesn't win, right? The bacteria that are present usually outcompete it and keep it under control. When those guys are maybe debilitated by antibiotics, that's when this one can overgrow. Or the case of Clostridium difficile can take up residency because all the other bacteria have been killed off by antibiotics. Because when you take antibiotics, they don't target just the microbe that's causing the infection. They target any microbe that antibiotic works against. So especially broad spectrum antibiotics, broad spectrum means it works over a large spectrum of organisms. As opposed to more narrow spectrum antibiotics, like say straight up penicillin G, plain old penicillin pretty much only works against gram-positives. So if you have a gram-positive infection and it's sensitive to penicillin, that would be your better choice instead of taking something like Augmentin, which will has amoxicillin, which will affect both gram-positive and gram-negative, and a drug associated with it. It's actually a combination. But when an animal bites you, you want Augmentin, trust me. It's a whole lot of nasty. And if a person bites you, you want some serious antibiotics too. Because I don't care what anybody tells you, right? Animal bites and people bites are the most likely to get really infected. Because mouths of dogs and cats are not clean, people. And they have sharp pointed teeth and they give you puncture wounds, which can create anaerobic environments, and really nasty bacteria can grow in that environment. People will crush the shit out of your tissue, too, which causes a lot of tissue death, which again will create an anaerobic environment. So who was it that Mike Tyson bit the other boxer's ear off? Yeah. Great, crazy people. Little kids bite too, by the way. Been there, done that. Everything in the mouth. So the next we're going to look at is complement system activation and long double-stranded RNAs. So basically interferon. So again, we really want to focus on what happens when these things are turned on. All right, we've already looked at toll-like receptor engagement. We're going to focus in now on complement, and then we'll look at interferon. So short video or animation.
Regardless of whether the complement system is activated by the classical, alternative, or lectin pathway, the activated complement proteins are involved in several important processes associated with host defenses, including inflammation, attraction of neutrophils, opsonization, and cell lysis.
So we talked about this earlier, right? Remember opsonization? Think tagging for destruction. And we saw this, like C3b was tagged onto the bacteria, and then the phagocytic cell had a receptor for it, and they grabbed onto it and phagocytized. Attraction of neutrophils and inflammation Remember, attraction, this is a chemotactic agent, right? Where C5a is released and the neutrophils are going to head towards it, right, to that area. Inflammation in general means white blood cells heading to that area. So these three are the letter number combinations that you need to memorize. The C stands for complement. The letter is in the order in which that protein was discovered. And when it has an A or a B, it means it's a fragment. So as you'll find out, these proteins that break into pieces, right? C3A helps in inflammation as well as C5A. C3B, how I remember this one is I remember B for binding. So remember, it opsonizes, it binds to the surface of bacteria and tags them for destruction. The several ones that come together to form the membrane attack complex, we're just going to refer to as the MAC complex, which stands for membrane attack. I'm not going to make you guys memorize all the ones that make up the MAC complex. We'll just refer to it by its structural name.
What did you say the number was? Like the 3 and 5?
So the C stands for complement. The number is the number in which the protein was discovered. If it has an A and a B, it means it's a fragment. So C3 breaks into part A and B.
Complement C3A participates in inflammation by inducing changes that cause the walls of local blood vessels to become more permeable. This facilitates movement of phagocytic cells from the circulation into the tissues. Complement 5A induces a directed chemotactic migration of phagocytes to the site of complement activation. Complement C3B functions as an opsonin. It binds to microbial cells, making them more susceptible to phagocytosis. Complement proteins C5b, C6, C7, C8, and C9 react together to form a hole in the cell membrane. When C5 convertase converts C5 into C5a and C5b, the C5b binds to the cell surface and combines with C6. C5b6 binds C7, enabling it to insert into the membrane. Binding of C8 is followed by binding of several C9s. The hole generated by formation of this membrane attack complex causes the cell to lyse.
Right, so it's these donut-shaped protein complexes forming by several of the complement proteins coming together. The important thing here is to notice it's inserting into a membrane, right? It's called the membrane attack complex, or MAC for short. And it literally physically can cause a lysis of the cells. So complement, when it becomes activated, it can actually lyse foreign cells. And then the other components, as you saw, can help with the inflammatory response, and then C3b binds, right, and opsonizes. So these are our important outcomes, right? We get inflammation, right, attraction of the white blood cells to the area to phagocytose. Phagocytosis is enhanced by opsonizing, right, by tagging for destruction. And we can actually lyse foreign cells by the MAC complex, the membrane attack complex. Right, so the important ones, C3A and C5A, right, these are going to help in the inflammatory response. C3B actually binds to the surface of the bacteria and allows phagocytic cells via their C3b receptor to bind and phagocytose these. And then the MAC complex, that donut-shaped hole. So why is it more effective against gram-negative cells than gram-positive cells? What do you know about structural differences versus gram-positive and gram-negative? Gram-negative has an outer membrane, right? So that's its outer covering, and what does the MAC complex insert into? Membranes, right? So gram-negatives are very susceptible to attack by the membrane attack complex and destruction. Where for the gram-positives, it actually has to travel, those components have to travel through the cell wall to get to the cell membrane to disrupt those cells. It can do it, but more often it's going to destroy those gram-negatives first. So then that's what it looks like in an electron micrograph. You see all the black holes are all holes in that cell by MAC complexes inserting into it. I'm not going to play this next one. It talks about, the different proteins, and they're actually numbered in the order in which they were discovered, not necessarily in the order in which they're activated. There's 3 ways we can activate this system. One is the alternative pathway, one is the lectin pathway, and one is the classical pathway. So the lectin pathway involves mannan-binding lectins. These are pattern recognition molecules, right? So again, we're looking at similar patterns. They detect the polymer of the sugar mannose, known as mannan. And this is very common in microbial cells, not our cells. And so when those pattern recognition receptors recognize mannan, they can activate this system. So this system has a common point, whether it's activated by the lectin pathway, the alternative, or the classical. They're all going to cause this splitting of C3 into C3A and C3B, right? So we keep breaking C3 into its 2 components. As we've already said, C3A is going to help with inflammation, right? Making those endothelial cells lining the blood vessels leaky. C5A is going to come later when C3s break down C5s into its components. C5A is going to specifically call neutrophils to the area. C3B, as we know, binds to the surface of bacterial cells and helps tag them for destruction by phagocytic cells. C5b is going to go on, if it can, and help in the formation of that MAC complex. Now, are all these things always happen? No, right? Sometimes C3b can't bind. Sometimes the MAC complex doesn't form. But pretty much all the time you're going to get an inflammatory response when you activate this system. I was trying to remember if I had to zoom in. So the alternative pathway, C3 is always breaking into its pieces. And when C3b binds, it leaves C3a all by itself. And so this is going to start that cascade of events, right? Where we're going to get more C3a and C3b. And we may even get the MAC complex formation. This is known as the alternative pathway because the classical pathway was actually discovered first. And what happens with the classical pathway is antibodies bind. And complement proteins bind to the antibodies. And that starts the cascade of events, right? C3 into C3a and B. And we may even get MAC complex formation. This is probably not what happens first. Classical is probably not what happens first because classical needs an antibody to start. Antibodies, right, are very large proteins that our adaptive immune response produces in response to a specific antigen, right? So if this is the first time you've seen this bacteria, do you have an antibody against it? Nope. So you can't do it the classical way, right? But again, this is the way we discovered first because it was easy to study. And so then, of course, the alternative pathway is referred to as the alternative because it wasn't what we discovered first. But in truth, the alternative is probably what happens first, right? The very first time you see a bacteria, alternative pathway or lectin binding is being activated, not classical pathway. But does it really matter? No, because just as long as it gets activated, we're going to get protection, right? But the good news is that we can activate it 3 different ways. Right? We don't have to rely on just one way of activating this system. But you're going to get similar outcomes each time, right? Hopefully you may get C3b binding. we have a way of stopping it from binding from our cells, right? We have regulatory proteins that stop C3b from binding, because then we would destroy our own cells, right? We would activate complement, and we get cell lysis and inflammation, and that would be bad, right? Microbes, on the other hand, normally can't stop this process. C3b binds and activates complement. There are some bacteria out there that have avoided this. You betcha. There are some of them that coat themselves in our regulatory proteins and stop the activation of complement. They use our own defenses against us. So again, we still get sick even though we have all these defenses, right? Some microbes out there have found their way past, or at least long enough to cause damage for us. Because we don't give up, right? They may get in, but usually we will not give up. All right, so this is an excellent video animation on interferon antiviral activity. So I am going to play this one for you guys.
When a cell is infected by a virus, the virus enters the cell and produces structures that are not found in uninfected cells. The presence of this viral material signals the cell to produce interferon. The interferon moves out of the cell and attaches to receptors on nearby cells of the same type. The cell that produces the interferon is unable to save itself. The virus replicates in this cell and then moves out to infect nearby cells. The nearby cell that already has interferon bound to its surface responds in several ways, including production of enzymes that degrade messenger RNA and prevent protein synthesis. Thus, a virus can attach and enter the cell, but completion of the viral replication cycle is prevented.
That's pretty cool, huh? So
Interferon doesn't save itself, it saves other cells.
yeah. So, oh, I don't have the drawing? What did I do with the drawing? Okay, so cell gets infected, right? So it's like you get wounded, right? You're going to scream, shooter, right? If you got shot. So everyone else around you knows that someone's shooting, say at the crowd. You may not be able to save yourself, right? If it's a crucial wound, you're going down. But hopefully you warned your buddies next to you, and they respond accordingly. So in the case of the cells of your body, they get prepared, right? Their friend right next to them just is going down, right? He's been shot. So they scream with interferon. Their friends acquire that, and they get prepared for the possibility that they may be attacked next. So they're going to produce, and if they do get invaded, they're actually going to be aggressive about this, right? They're basically going to attack the shooter, right? Because they're going to produce enzymes that will destroy that virus's messenger RNA. Now, there's a problem with this, though, right? What's going to happen to you if you attack a shooter, probably? You might get shot and you might die, right? But you may stop him and save all your friends around you, right? What happens with these cells that get invaded? They're doing the same thing. They're basically giving up themselves to stop the virus. Those enzymes that they produce will kill them as well. It will destroy their own messenger RNA once activated. But does the virus replicate in them? No. So then we stop the spread of that virus, right? We're a multicellular organism. Can we afford to lose a few thousand cells? Sure. Right? Remember how we talked about the flu, how just one virus gets in one cell? Right? So a whole lot of our cells die in the beginning until we get this under control. Right? Until we can stop that cycle. That's why the flu sucks so bad. But we can win, right? Well, this is one of the ways that we win, right? Is those cells that get infected that know about it, they destroy the invader and stop its replication, stop its spread. So it makes sense, right? And for some drugs, from some virus treatments, they would actually give the person more interferon so that more cells are aware that a virus is present. Get prepared. In case you get infected, you need to destroy the invader. That's your job. Make sense? Right. Right. So when they receive the signal, right, they're going to make proteins and get ready, right? So that when the virus actually comes in, then they activate those proteins.
So it's antiviral proteins that are inactivated.
Mm-hmm. So interferon causes neighboring cells to express inactive antiviral proteins, right? So they make the protein. So they get ready, right? In case they're next. When the virus goes in, it activates those proteins. Those proteins attack the virus and digest it and stop it from replicating. But in the process, that cell also dies, right? And it goes through programmed cell death, right? Apoptosis. What's unique about apoptosis? It doesn't cause inflammation, right? So we're not going to get an influx of neutrophils in this situation, usually. The macrophages that are resident cells there, they're going to clean up the debris of these dying cells. And they're not going to call in reinforcements. They usually can handle it themselves. But sometimes with some viral infections, you will get a rash, right? That is inflammation. Right? Sometimes when you have a virus infection, you will get inflammation. Especially if this is failing. Right? Because again, every protection we have, there's something out there that knows its way around it. So the last is fever. So what's the benefit of fever? Why do we increase our core body temperature? Why can heat be damaging? It denatures proteins, right? So if the microorganism, if we get too hot, we could denature their proteins and shut them down, right? Plus chemical reactions. We were doing this today, right? Hot water or cold water for if you're trying to dissolve something. Hot water, right? Right? We were trying to get hot water today. That was fun. I don't know if you guys have been in the biology labs. I haven't until recently really been in the biology labs. They have these stupid hoses from the sink. If you don't hold it, it squirts everywhere. That's so good. I'm going to buy some tubing, plastic tubing at Lowe's tomorrow morning. Fix that problem. Because it was right next to my computer. Scared the crap out of me. Electronics and water. Right? So, same thing here, right? So, we're getting hot, right? So, chemical reactions are going to happen faster. We could denature the bacteria and we could kill them. These sound great, right? Heck yeah, why don't we have fever all the time? I mean, I'm just going to walk around at 101 degrees all the time. Why not, right? Be antibacterial all the time. Is that good for our cells? No, right? Being hot for a long period of time, we could denature and harm our own cells, right? And why would you actually want to be faster? Well, because you've got invasion going, right? You're not going to slowly walk up to the bad guy. You're going to run over and disable him, right? As fast as you possibly can. So heating up makes sense. We can move faster, right? And we may get too hot for them to grow effectively, right? We might denature them and destroy them. So we can control fever, right? We can turn it on and we can turn it off. Those would be our own endogenous cryogens, right, that induce fever. The problem that exists is that some microbes produce substances that cause the fever response to be turned on in our bodies. So these are exogenous cryogens, right? These are produced by the microbe, not by us. Bacteria There's lots of bacteria out there that do this. So we get hot, it doesn't hurt them, but it will hurt us. So usually like when you have a kiddo, right, who spikes a fever, and this always happens at night, right, and you end up in the emergency room. The fever just keeps going up and up and up, and it's not going away. Even like when you give Tylenol and a Motrin, right, it just comes right back. That's usually a pretty good sign that they got a bacterial infection. In the case of my son, that was in his ears, right, when he was young. And he obviously has a very high pain threshold because he never complained of his ears hurting him. But they were infected pretty bad. Little kids, unfortunately, their ears, canals, right? So your stationary tube that drains into your nose is not very pitched. It's almost horizontal. So it doesn't drain real well. And then if you have problems like allergies and stuff like I do, my son does, the adenoid, which is the tonsil in the back of your nose, can swell too, and that can block the stationary tube, so keep them from draining. So you got fluid building up. Microbes that go up your nose can sneak up the stationary tubes and get in the inner ear, and they can grow in there. And then that creates pressure in the system as well, which is usually why the children have the ear pain. But again, you know it's bacterial when you're spiking that fever and it's not going away. For him, he ended up with tubes in his ears when he was 2 years old, and they decreased the size of his adenoid. And then we also realized it may be related to allergy and put him on allergy medicine. Basically, problem gone. And then as he grew, as your head grows, you also get a greater pitch to your stationary tubes, and they drain better into your nose instead of being more lateral. So that's the fun of the anatomy, right, of our bodies sometimes, and the good old microbes that can also cause us to have fever. And this is why this is not something, you just take Tylenol and you go, oh, well, I'll go see the doctor. If it's not going away, right, you probably have an infection that needs to be dealt with. Very strong indicator. Now, do you have fever sometimes with viruses? Yeah. Usually tends to be intermittent. Goes up, comes back down, goes up. Doesn't prolong and continue to increase normally. All right, I just want to get through a couple of slides of this one because it'll be a good intro to the ones that you guys are going to be watching, okay? So I promise only 15 more minutes. All right, so we're done with innate. Now we're going to go into adaptive. So this is the component that changes over your lifetime and continues to adapt. This is where your antibodies come from, right? That are specific to specific invaders that have come inside of you. So this is the most effective, right, because it's very targeted, right? We're specifically targeting specific invaders, specific viruses, bacteria, fungi, parasites. The problem with this system is that it can take a week or longer once you've been exposed the very first time to that pathogen to mount a response. So in the meantime, guess what he's doing? He's taking up shop and he's eating away at you and growing and growing and growing and potentially producing toxins. So for some of the really bad bugs out there, it kills you before your adaptive immune system gets online. If your innate can't take care of it, right, can't keep it under control, it kills you, right? Tetanus can kill you because the toxin that it produces can kill you in the matter of hours. And you need how long before you can mount a response specific to that toxin? Not hours, a week or longer. Make sense? So innate's got to keep you alive long enough that this can kick in if the innate can't eliminate it on its own, right? Your innate is pretty powerful, right? Don't discount that, right? Those phagocytic cells are pretty good. But if they can't take care of the job and they need additional help, they've got to hold it off, keep you alive in time for this system to be online. The good news with the adaptive is it has memory built into it. So notice we said first exposure. The first time you're exposed to it and it mounts a response, it remembers what it did. So the next time around, right, when you remember something, you do it faster, right? It responds that much faster because it remembers. Vaccination exploits this ability of memory. So what we do is, in a relatively safe way, is we give you that first exposure. So when you get the flu vaccine, they're giving you a virus, right? Several viruses, potentially what the ones are going to look like that are coming around this year, right? Because the influenza viruses mutate very fast. That's why we have to do a new vaccine every year. Unlike measles, right? They haven't changed that vaccine in forever. That virus hasn't mutated, right? So that vaccine is still good. We can give it to you, but it's going to take, once we give you that shot, 7 to 14 days on average for your immune system to mount a response, to make antibodies, make cytotoxic T cells that specifically recognize that virus. So you can't go into Walgreens tomorrow, get your flu vaccine, and walk out and think you're fine right then. So there's tons of people who are like, I'm never going to get the flu vaccine again because I still got the flu. Well, one, again, you're buying a lottery ticket, people. There's no guarantee it's going to work. But there is a chance it could work, right? Just like when you buy a lottery ticket, there's a chance you could have the winning ticket, right? This is why you buy it. Still with me? Okay. But also, you know, you kind of have to wait for the drawing for the lottery. You also got to wait for that vaccine to work. So unfortunately, in the meantime, you could be exposed to that virus, and yes, you could get the flu, even though you've got vaccinated, and even though maybe that vaccine could have helped you, but the timing was bad. Some people experience fever after getting the shot, feel kind of weak and sick. Right? That's because your immune system is working. It's doing its job. Some people have very active immune systems, and they feel really crappy. Others, they don't even notice it's working. It really can vary from person to person. So those are the things that you have to keep in mind as far as vaccination goes. This is a molecular specificity. That antibody, those T cell receptors, right down to the amino acid sequence differences in the proteins associated with these pathogens that you're recognizing. So antibodies can specifically detect specific proteins, right? If we put a whole bunch of proteins in a test tube, the antibodies will only bind to the ones that they recognize. It's very specific. This system also has to be able to detect when you have healthy cells, right? You don't want to attack your own healthy cells. You don't want to attack yourself. But it can attack yourself potentially when it's not healthy, right? When you have cancerous cells or cells behaving badly is what I like to call them. Right? This system can detect that to a certain degree. But of course, more often than not, right, it's our primary defense about specific pathogens that we've seen before. So when someone says they're immune, that means that you have this protection, you have this memory, right? Either you've had the infection, like for me, I had chickenpox. Oh boy, did I have chickenpox. I had it when I was 4 years old. And when I was a little kid, there was no vaccine. So your mom brought you over to somebody else's house who had chickenpox so you could get chickenpox before you went to school. Right? My son got the chickenpox vaccine, because in 1995 they came out with the vaccine. So he doesn't have, hopefully will never have to deal with getting chickenpox. Yeah. You've been vaccinated? Cool. Lucky you. Those of us that went through it, Kat's like, oh yeah. It was horrible. I still have scars from the ones I picked off my knees. Yeah. It wasn't fun. And that's a whole nother ball of wax, because whether you've been vaccinated or you had it, you are immune, but you also have that virus in you, and it can reactivate and cause shingles, right? Unlike people like, was it measles or mumps? I can't remember. I think it was measles. Measles was widespread, and some people died from that. But some people, their immune system was able to take care of that viral infection, and they survived, and they have immunity from it. So before that vaccine came online, there were some people that were naturally immune, right? Where they got natural exposure, they had the infection, and they have immunity that way. Right? So although they were never vaccinated, if you were going to do an antibody titer on them, if you were to take a blood sample and figure out the concentration, they have quite a bit of antibodies against that virus because they had the actual infection. We can't tell the difference unless, I don't know if they do like really highly expensive, but like if they do a titer on you, like if you're getting ready to go to nursing school and stuff like that, nowadays they don't really just even accept just vaccination records. They want titers. They want to know that you have those antibodies in your system. Is that me that keeps doing that? I guess so. Those antibodies in your system. So they're going to do titers on you. When you're pregnant, they do titers to see your level for chickenpox because that can be passed to the baby, and rubella in measles. German measles is rubella, and regular measles. They want to know what your protection is, and if you're low, they're going to caution you to be careful. And then after you given birth, after a certain period of time, they're going to suggest that you get revaccinated against those viruses. So, as I said, that's right down to molecular level. So here's our little cartoon guys again, right? So it's receptors on the surface of our cells that are detecting. And we have two types of protection within adaptive. We have a cellular response where cells are attacking other cells. Or we have what's commonly referred to as an antibody response, but typically known as your humoral immunity. And again, this is dependent on the type of pathogen we're dealing with and what cells are going to be involved in your response. So antibodies cannot enter into our cells. So this is kind of an extracellular response. So whenever something is outside of our cells, it's going to help tag it, and it's going to opsonize it. It's going to tag it for destruction. It can even activate complement, right? Antibodies do amazingly awesome things because, again, they specifically bind to specific pathogens that you've seen before, right? Either by natural infection or vaccination. Other responses, your cellular response by your T cells, is if something gets inside of your cells, right? So when the virus is inside or the bacteria is inside, we have a way of detecting when those cells are infected and to be able to destroy those cells. All right, so we're going to watch an animation on that.
Activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen. B cells also come in millions of different types, each able to recognize a particular antigen. When B cells become activated by T helper cells, they differentiate into plasma cells. These plasma cells become antibody-producing factories, flooding the bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the B cells do not turn into antibody factories, but instead become memory B cells that may survive for several decades. Because of these memory B cells, the secondary immune response to a future infection by the same pathogen is swifter and stronger. This powerful secondary immune response is what gives immunity to some diseases after you have had them once or after you have been vaccinated.
So what they don't show you there is T cells, when they become activated, some of them go and do their job, but some of them also become memory cells as well. So for your T cells and your B cells, you always produce memory cells when they become activated. And then again, this is the important memory component, that once this happens, then you're going to remember it and you're going to respond that much swifter the second time, or the third time, or fourth, whenever you see it again. So there's one mistake in this animation, which is unfortunate, because it's pretty good. So notice here, right, these interactions. So it's presenting the antigen. There's actually particular molecules that we have associated with our cells. There's two classes. They're MHC class 1 and MHC class 2. And this is why they communicate. I almost consider it like a secret handshake. This is why these cells can communicate with each other, is because not only is this cell recognizing the antigen that that cell is presenting, it's also recognizing this receptor, right? So, you know, it's their special handshake. They recognize each other. And then they're communicating, oh, yes, I recognize this particular antigen. So then they're going that one step further, right? They're communicating that, and they're causing something to happen. So T cells have T cell receptors, right, that recognize specific antigens. So you make billions of different T cells. Each has a different receptor on it. And it's just like playing the lottery. That information is encoded in your DNA, but it picks different pieces. Just like you pick different numbers for the lottery, it picks different pieces to be able to make that receptor. So each T cell is unique. But when that cell becomes activated, it makes copies of itself, right? So then you have a whole bunch of them that recognize the same antigen. And again, this is a specific interaction. This cell can only talk to those types of cells because those receptors recognize each other. Still with me? So that's MHC class 1 because it's on all of our cells. Antigen presenting cells like this macrophage back here, that would be MHC class 2. And that's why it'll talk to helper T cells, right? That's their co-buddies, right? The B cell, what they're not showing here is, see, it has the receptor as well, where it can present antigen, and that's what they're not doing correctly in this animation. The antigen should be over here, not attached to the antibody. It should be on the MHC class 2. And this helper T cell should be interacting with that. But B cells have their own B cell receptor, and again, it's unique to the antigen. So notice it's able to bind to the antigen, and then it will actually engulf it, break it down, and present it on MHC class 2. that will allow it to talk to the helper T cell, right? And it's going to cause that B cell to proliferate. And as I said, the B cells then proliferate and they turn into memory cells and plasma cells. The reason why they're called plasma cells is their cytoplasm increases exponentially. Their rough endoplasmic reticulum gets enormous. Because why is it called a rough ER? What makes it rough? Ribosomes. What do ribosomes make? What are antibodies? Proteins that are secreted. Proteins that are secreted are made in the rough endoplasmic reticulum. That's why when a B cell turns into a plasma cell, its cytoplasm gets enormous because it's going to produce and secrete, as they show you here, antibodies into your tissue fluids, into your blood, right? All these guys are released. There they are. So that's just in words what we were talking about earlier. This is an excellent schematic that I really like. It's relatively simple, right? And shows the different components, right? So we have humoral, which is antibodies. So that all comes from B cells being activated, right? Turning into those big plasma cells and antibodies being produced. Where your T cell response, right, is your cellular response. So here you have cytotoxic T cells that will go and attack infected cells. And will also activate helper T cells, which, as you saw, need to help out the B cells sometimes in their activation. And they can also help macrophages become those more powerful killers that we talked about. So when the macrophages engulf it and they present it to the helper T cell, right, they can be like, oh, yeah, I know that bad guy. You really need to do this next to destroy him, right? You better make some peroxidase because that one really gets him, right? All of this starts with, notice our innate immunity. Things like the dendritic cells or the macrophages actually engulfing the pathogen and presenting it to the helper T cells or the cytotoxic T cells, or both, y'all. Because think about this, where do viruses get? Inside of our cells, right? So what type of response would we want? We want to be able to kill those cells that are infected. Which one of these pathways is going to do that? Cytotoxic, right? The cytotoxic T cells are going to recognize the infected cells and kill them. But are the viruses always inside of our cells? No, they pop out and they've got to travel outside to get into the next cell. So if we also produce antibodies against them, we could bind to them before they attach to us and phagocytize and destroy them. This is called neutralization. When an antibody binds to a virus before it binds to us, we basically neutralize it, right? It can't get into our cells. So when you get vaccinated, you want both of these avenues to happen against a virus. You want a cellular response and you want a humoral response. So when I first graduated from undergraduate in Massachusetts and I moved to Louisiana, I didn't get into LSU vet school because I was from out of state. They have only a few spots, right? So I got a job working at LSU vet school in research, doing leprosy research to establish residency and reapply. Of course, when I got called for my interview, I decided I didn't want to go to vet school anymore and went and got my master's degree at Southeastern instead in immunology because this stuff is cool. But when I'm studying leprosy, this is a pretty fascinating disease because, again, how does your immune system work? Where is this information in your body? Where is it, Annie? Where's all your information in your cells? It's in your DNA. It's encoded in your DNA. So with leprosy, you'll find that it tends to run in families because, again, their immune systems are all the same or similar, right? Because they're all family. They also live in the same area, so they're probably exposed to the same pathogens, right? And so they have the ability to get infected because they have a defect in their immune system, that doesn't kill the mycobacteria, right? And they're all exposed to the same stuff. So that's why you have that phenomenon where it seems like it runs in families. No, they're not cursed, right? It's geography and it's DNA, right? For some of them, unfortunately, their body just goes antibody response. This is an intercellular bacteria. What good is an antibody going to do if the bacteria is inside your cells? Not going to help you, is it? It can grow inside your macrophages. It'll infect the Schwann cells that surround the myelin sheath of your nerve cells. And it actually prefers the ones in the periphery because they like cooler temperatures, right? And at your extremities, your ears and your hands and your feet, that's where you're cooler. So the bacteria migrate there. And they grow inside the cells. If you're just producing antibodies, it's not going to give you any type of protection because the antibodies don't get inside the cells, right? So it can actually, for them, it'll grow uncontrollably in their lymph nodes, in their spleen. It'll cause their spleen to burst, right? If not treated, it can overgrow and kill them. Other people that get the deformities and stuff like that, especially when the bacteria head to the extremities, they're completely a T-cell response, is what we call tuberculoid leprosy. And so their immune system can kill the bacteria that are inside your macrophages, inside those Schwann cells. The problem is it also damages those nerve cells that are infected, right? They're chaperone cells that are infected. Can we repair that damage? No, right? So they'll, and again, if we catch it, we can also give them antibiotics, right? We do have antibiotics that will kill Mycobacteria leprae. So we can kill the leprosy, right? We can kill the Mycobacteria leprae, but we cannot repair the nerve damage that could happen for some people who have that cellular tuberculoid response. So those people that you see with the deformities and stuff, right? That's again due to the destruction and then a lot of times secondary infection. So there was a patient, and there's actually a guy who still lives there. He came originally from the Philippines that's still at Carville in Louisiana, right outside of Baton Rouge, in St. Gabriel Parish. There was the last leper colony in the United States, shut down around the 80s. But there are some people, they never lived anyplace else, and he has severe deformities. His fingers are just nubs, right? Because this was before the treatment came online. His toes, he's lost most of his toes. And he's told a story about a patient that went into another patient's room and he stuck his hand down on a hot plate. And the only reason why he knew his hand was burning is because he smelt it. He did not feel it at all because of the nerve damage from the infection. So they have to be really careful, like diabetics have to be careful with wounds to their feet and hands. Because if you don't notice you've gotten wounded and it gets infected, right, you could lose a limb because of it, right? Because they don't feel it. They don't feel it at all because the nerves have been damaged so extensively. Now, they've done some really great research in that, in nerve help and nerve repair, which has also helped people with diabetic neuropathy that happens with diabetes as well. But this is a really fascinating, it's like, well, why are some people just producing antibodies and why are some so aggressively T-cell response? And then we've actually found that there are people that are right in the middle, they do both. And then some that head a little bit more towards antibody response but still have a slight cellular response. And others that do a very large cellular response and a little bit of antibody response. But in the case of this type of disease, you really want a cellular response, right? Because these are intercellular bacteria. The antibodies really aren't going to help you. So when I worked in research, we purposely infected bacteria, the leprae, into armadillos because they get the infection just like we do. And we would grow it in the armadillos. And they have they're mammals, they have a cooler body temperature than we do. So the Mycobacteria just loves them. So we would, once they succumb to the infection, I would monitor their progress by taking samples and monitoring their antibody levels, right? I would look at their titers for the antibodies against leprosy. And we would test them as they came in. We would do a skin test, just like you do a skin test for TB. We would inject them with mLepre proteins into their skin. And then we would biopsy it out. And then we would stain it, and they would slice it and make slides. And we would look at it under the microscope and see what happened. If T cells, and you can even see there would be a bump like you do with the tuberculoid test, that would be a lot of influx of T cells and macrophages to the area. We didn't want that. We didn't want a cellular response. We wanted a nice flat area, so we would actually tattoo the area where we did the Lepperman test, so that when we went back, we could biopsy out exactly where we put the bacteria in there. If it was nice and flat, that was a pretty good sign. And under the microscope, if we didn't see any cellular invasion, then probably they would get lepromatous leprosy, which means that they would do strictly pretty much an antibody response to the bacteria. So then we would purposely inject it into them, into their veins, the leprosy, a live inoculum, and give them leprosy. Because you cannot grow mycobacteria in artificial culture, because it's an intracellular bacteria. You'd have to basically do tissue culture, which they've tried to do. When they're trying to grow actually really large numbers of bacteria, they actually inject it into the nude mice foot pad. So into the foot pad, again, extremity cooler region. They inject it into the foot pad. Nude mice don't have any hair because they have a non-functioning thymus. Guess where your T cells get educated and why they're called T cells? In your thymus. These guys don't have a functioning thymus. They don't have functioning T cells. They don't mount a cellular response. So the bacteria can grow to huge numbers in the foot pads, and then they're able to take that tissue and get the bacteria out to be able to do research. Kind of crazy, huh? But it's pretty fascinating when you think about like that, you know, for one particular bacteria, how different people are, and even the armadillos, right? How that code in your body of which way you're going to go. It's pre-programmed, right? But can we change that program, right? There's still so much we don't know. It's insane. Like, immunology, you guys are barely scratching the surface. Our research is barely scratching the surface of what happens in our bodies as far as the immune system goes. It is definitely uncharted territory. All right, y'all. Chart some safe territory home. Make sure you do those bonus videos, right? Because that's going to be paramount to us successfully completing this stuff.